Thank you very much for inviting me to this wonderful meeting. I was looking forward to join it in person, but unfortunately for family reasons, I'm not able to join it in person. But nonetheless, I'm delighted to be able to share with you some advances from my group. And I would like to thank very much Ryan Kelly and Theo Alexander for organizing this meeting at such an exciting time for the field and bringing together in one meeting uh, people developing mass spectrometry technologies for single cell analysis. It's a wonderful initiative that I would have loved to join in person. Uh, Starting with the question as to why do single-cell proteomics, one obvious answer is the enormous functional and molecular diversity of the cells that make up our tissues, as illustrated here with the cancer cell encrusted by T lymphocytes, and similarly stem cells differentiating into a variety of cells with obviously different morphology. But in addition to these very apparent differences of cells that can be sorted out and separated into bulk samples, there are also many cases where cells from the same type, cells that look the same way morphologically, are functionally and molecularly different, as illustrated with cells primed to develop drug resistance. And I'm going to give you an example of such model system studied with single-cell proteomics, or macrophage polarization, exhaustion of immune cells, cellular senescence, and a number of physiologically and pathologically very important processes that are not easily discerned from the morphology of the cell and very much benefit from the ability to perform unbiased molecular analysis. And of course, this analysis can be performed by RNA sequencing methods, but as useful as these methods are, they fail to capture important biological aspects, as shown here with p53 transcripts measured with two different methods that mostly agree with each other, but show very different dynamics compared to the protein levels measured again with two different methods that agree within each other, but differ from the transcript abundance. And this difference between p53 transcript and protein level is relevant for its function, as we showed by doing single-cell analysis of p53 and identifying that the p53 protein level is predictive of its transcriptional activity, while the transcript is not. And we are very interested in being able to use accurate high-throughput protein measurements to be able to distinguish between different classes of models based on methods that use very few assumptions, as described in these references here at the bottom. Of course, doing single-cell protein analysis by mass spectrometry faces many challenges, as summarized in this cartoon here from about a year ago, and nonetheless has already dated, which is wonderful demonstration of how quickly our field is moving forward. And there have been a number of contributions from different groups to this advance, including very compelling demonstrations of the importance of peptide separation, narrow bore columns, improved ionization. Ryan Kelly has published a number of very compelling results in that direction. Our efforts have focused more on the side of experimental designs, data acquisition, and interpretation, and that is going to be the focus of my presentation today. I'll give you a very brief update on our approach to doing single-cell sample preparation. We are excited to increase the throughput and decrease the background of preparing samples from single cells. And therefore, we have developed a method specifically aiming to do that. Specifically, it involves preparing the samples on the surface of a flat microscopic glass slide, where each cell is going to be prepared in a small nanoliter droplet. And we like this approach because of the flexibility to print these droplets in any geometry that we want, and because of the ability to increase the density of droplets on the glass slide, and therefore be able to prepare thousands of single cells simultaneously, using a completely automated approach that results in pulling our multiplexed samples into a multi-well plate that then can be placed into the autosampler of an LCMS system and analyzed automatically. What we like about this method is that it allows us to achieve very high efficiency of delivering proteins to mass spec detectors, 95% of the efficiency achieved by handling samples composed of thousands of cells. And we also like very much that the background is low. Background has been challenged for us with multiplex proteomics for a long time using different sample preparation methods. And in this case, we see that if we have a droplet in which we don't position a single cell, but we put all other chemicals, and that droplet undergoes all other processes as the droplets with single cells, that droplet has no detectable intensity for the mass spec instrument, as shown here with the negative controls, and we find this an important advance. We also like that this method results in samples that have very low abundance of +1 ions eluting during the active gradient. The majority of the ion current that we measure comes from +2 and +3 charge ions that are likely peptides. And there's this wonderful observation that has informed many of our efforts on the side of data acquisition and interpretation, which is that if we inject a single cell into the mass spec instrument, as many of us have done, we can detect tens of thousands of peptide-like features. And many of those peptide-like features are being represented by hundreds or even thousands of copies. But of course, detecting the peptide-like features is insufficient. We would like to identify what are the corresponding amino acid sequences, post-translational modifications, and quantify them. And in some cases, quantification cannot be done simply by detecting the peptide-like features. So we have developed approaches to take advantage of this high sensitivity of the instruments and maximize the number of peptide features that we can interpret biologically. And one of the challenges of analyzing all of these peptide-like features by MS2 methods is that for single-cell analysis, to be able to determine the sequence, we need long accumulation times, and also to be able to do accurate quantification, because if we count only a few copies of an ion, quantification is going to be noisy with low precision. So when we go into the realm of accumulating ions from single cells for hundreds of milliseconds, then we can analyze only 10,000 or so peptide-like features per hour of active gradient. And then we don't even assign sequence to all of them. Based on tandem spectra, we maybe sequence half of them. So what we wanted to do was to make sure that the features that we identified, that we spend our precious instrument time on, are those that we are biologically interested in, and that we can maximize the fraction of those features to which we assign confidence sequence. To do that, we used the real-time alignment feature of MaxQuant to prioritize the precursors corresponding to biologically interesting information, such as post-translationally modified peptides or peptides from proteins that are biologically relevant for our investigation. And the other very important aspect that we did with this approach is to develop a list of priorities where, when we operate at full duty cycle, the instrument may detect all of the peptide-like features, but may not have time to analyze all of them, which can result in low data completeness because the instrument simply did not have time to send some peptides for MSMS analysis. So to avoid this problem, we assign priority tiers where we explicitly determine what is going to be the order, the sequence in which different sets of precursors are going to be analyzed. And of course, we can take advantage of the ability to accumulate ions from some particularly biologically relevant precursors for a longer period of time. So how well does this work? We compared the data completeness achieved with max quant life without prioritization in gray or with prioritization in red. And we see the expected result that without prioritization, max quant life doesn't result in high data completeness because operating at full duty cycle time at all times means that some precursors simply don't get to see the light of day. But with prioritization, this problem is resolved without decreasing the coverage of proteins per single cell. In fact, we see a small increase. And if you look at what the instrument does, you see that it implements exactly the logic as you would hope it does. We detect virtually all precursors on our inclusion lists. From the high priority ones, virtually all of them are sent for SMS analysis. From the medium priority, only about 60% are sent. From the low priority, only few of them are sent. And with this approach, we see that there is high data completeness when applied to analyzing single cells. And this is particularly striking in terms of gain increase for challenging peptides, as demonstrated here with a set of 1,000 peptides that are particularly difficult to identify consistently in single cells. We see more than 2 to 3-fold increase, and ultimately we get to about 94% data completeness. Now, do we achieve our aim of assigning sequence to all analyzed precursors? We get close to 90%. And what about the remaining 10, 12%? Those 10, 12% correspond to precursors that generate very few peptide fragments, and therefore they don't constrain robust sequence identification. But that can be improved further, especially if one uses a more modern instrument. In this case, we used QExactive Basic with a very narrow quad isolation of 0.5 Thomsons, and we know that this is very difficult to achieve on this instrument. So because of this much higher rate of assigning sequences to analyze precursors, we see about doubling of the number of peptides and proteins that we can quantify in single cells. And we also see that there is this whole range of low precursor abundances that now become quantifiable, as shown by this red distribution of precursors quantified by prioritization. And they're not quantified when using shotgun analysis, shown with the blue distribution. But this is about detecting peptides. What about the quantitative accuracy? One of the challenges of this approach is that we still use isobaric mass tags, and we are still potentially affected by co-isolation, potentially significantly. One thing that prioritization helps with is that we can more consistently analyze elution peaks close to the apex, and that reduces co-isolation. And by using the lower isolation window, smaller isolation window, we can further reduce co-isolation. So we observe that when we spike in synthetic peptides into single cells at levels comparable to the abundance of the single cell proteins, the quantified ratios for these peptides agree quite closely with the spiked in ratios. And we were quite pleased to see that pSCOPE is able to achieve this accuracy, not only higher sensitivity, but also good accuracy and very high data completeness. So with prioritized scope, we are able to analyze peptides of biological interest, but we don't analyze all of the available peptides. And the obvious approach to analyze all of the available peptides would be to use data-independent acquisition, which can at least collect the fragments of all precursors. Interpreting them is whole another matter. But we were very interested when using data-independent acquisition to also be able to multiplex multiple samples at the same time. And when we do that, we wanted to make sure that proteome coverage doesn't decrease and quantitative accuracy is high. Essentially, we were inspired by this possibility that if we are able to analyze in parallel both samples and peptides, we can achieve a multiplicative gain in throughput. And Jason Derks, shown here in this picture, who is with you at this wonderful conference, and you can address your questions about plexDIA to him directly. He also has a poster with new developments that I'm sure he'll be happy to share with you. So Jason Derks wanted to test very rigorously if we can achieve multiplexing with plexDIA without reducing proteome coverage or quantitative accuracy. So he prepared samples of mixed proteomes of different species that were either analyzed separately with label-free DIA, or they were labeled with non-isobaric mass tags, then combined and analyzed together as one sample. And what Jason observed is that the number of quantitative data points increased in almost linear proportion to the number of the mass tags used, as shown here. And he also observed much better overlap of the proteins that are consistently quantified in every single sample. So that resulted in being able to quantify more proteins when using plexDIA across all 3 samples and when using label-free DIA, while using 3 times less instrument time. And more importantly than just getting more data points is that these data points are accurate and precise, as shown here by the comparison of the measured ratios and the mixing ratios. We worked very carefully and rigorously on making sure that as we propagate peptide sequences within a set, we can do this reliably with empirically, not computationally, but empirically determined false discovery rate, which in this case, Jason accomplished by having one sample that contains proteomes from 2 species, such as yeast and E. coli, and another sample that contains only yeast. And then we made sure that our algorithms are such that when we have this sample that contains plenty of yeast proteins, lots of precursors, they're correctly assigned to yeast proteins and not to bacterial ones. And this works very well at both MS1 and MS2 level. Here is how applying plexDIA to single cells looks like. We are able to do quantification at both the precursor level and at the fragment levels. And we are able to quantify a couple of million copies from proteins per single cell, with the median number being about 30 copies per precursor or protein from single cells. This ability to have multiple scans during the elution of the precursors and their fragments allows us to obtain multiple estimates for relative abundance of a peptide between individual single cells at both MS1 and MS2 level. And the agreement between these estimates can inform the level of confidence that we can have in the data. And zooming out a little bit from showing you raw data, when we compute relative quantification ratios of proteins between 2 different cell types, PDAC and Q937 cells, from either single cells or bulk samples, we see very good agreement. And what I want to emphasize here is the dynamic range of variation that we quantify these relative protein fold changes is about 1,000-fold, something that is very difficult to achieve with the isobaric mass tags. And seeing that I'm running out of time to tell you about the biological application that Andrew Leduc developed, I'll just quickly skip and show you one technical result of that, where Andrew discovered the existence of those clusters of melanoma cells that turned out one of them is primed for drug resistance, the other is not. And he was able to use these 2 methods that use very different quantification bases, one using isobaric mass tags, the other using precursor and fragments at MS1 and MS2 level quantification, to consistently identify those clusters. And then when comparing the relative quantification of proteins between these 2 clusters, again, of primed and not primed melanoma cells, we see also good quantitative agreement between biological replicates and methods. And we think that this approach of comparing results from very different methods is a good one as we go forward in establishing results on solid foundations. I'll skip through these very interesting results. They're published. You can read the preprint because I don't have time to go over them. Andrew discovered a very interesting gradient of protein covariation within a cell type and within a cluster, not just within the cell type, but within the cluster of non-primed cells, which we're very excited about. I want to finish by saying that all of the data that I talked about and much more are available for you to download from this website. And we think that there is tremendous opportunities to further increase the depth of protein quantification, including post-translational modifications, and the throughput, as discussed in this perspective here referenced at the bottom. And what is wonderful is that this potential is increasingly being appreciated by funders. And we have been fortunate to be funded for establishing a large-scale technology platform as part of a new research initiative, New Research Institute in Boston, for advancing single-cell technologies and applying them to flagship biological projects focused on Alzheimer's disease and biology of aging. There will be a more formal announcement of this very soon. But for those of you who are interested and would like to join our efforts, we'll have many positions with generous funding and I think very exciting opportunities. And as more colleagues beyond the group of initial pioneers are beginning to use single-cell proteomics, we got together and developed a set of guidelines of what we think are good principles for going forward. We worked hard to make this useful to the entire community, and many of you helped. Ryan Kelly is one of the people who helped with compiling these guidelines. You can find them from this URL, and you can also contribute to the next iteration of best practices for single-cell proteomics. And I know that there would be a lot of exciting exchanges happening at this conference, but I also know that as the field is moving very fast, 7-8 months from now, there will be a lot more new results to discuss. And I hope that the 6th Single-Cell Proteomics Conference in Boston is going to provide a venue for those discussions. And I would like to welcome all of you who are interested to join us to do so. This is the team of people that made all the results they shared with you possible. In particular, Jason Derks led the plexDIA work. He is with you, and do ask him more about the latest advances on that work. Andrew Leduc developed the droplet sample preparation, nPOP, and did the melanoma work, for which I did not have much time to tell you. And Gray Huffman developed the prioritized analysis, pSCOPE. I would like to thank the funders who made our work possible, and I'll stop